Hi everyone, my name is Savani and I will be talking about human gene therapy. So, what exactly is human gene therapy? This is a scientific technique that modifies a gene in order to treat or stop certain diseases or conditions. In other words, it allows for the correction of defective genes responsible for disease development. So, to cure a certain disease, this therapy locates the abnormal gene and introduces new gene into the body. And there are three main ways to do this. This is replacing a disease causing gene with a new healthy copy, or inactivating or knocking out the gene that isn't functioning properly or it's not doing its job. And then lastly is introducing or inserting a newly modified gene into the body to help fight the disease. Now, there are two types of gene therapies. The first one is somatic, and the second is germline. So, for somatic, this is when the modified gene is inserted into somatic cells, which are blood cells or bone cells. And once it's in the cell, it's contained within it and it doesn't affect the surrounding cells. And since it doesn't affect the surrounding cells, it's not inheritable for future generations. And somatic gene therapy is much more common than germline since it has been researched and more reliable for uses. Now, germline gene editing, this is the modification of the much earlier stages of development. So this is in the reproductive cells like the sperm and the eggs and the ova. And once it's in the sperm or the ova, it can affect the surrounding cells. So due to this, it makes it inheritable and it's passed down from generation to generation. And since it's such an earlier development of inserting genes, it has brought up a lot of ethical and societal issues. Now, there are multiple delivery methods. The first one is in vivo, and this is when you directly deliver a modified gene into the patient. Whereas ex vivo, you take the genes out of the patient and then you modify them, then transfer them back into the patient. But in order to transfer the desired genes into our target cell, a carrier or vehicle is needed to do this. And these vehicles for gene delivery usually have vectors, and these vectors are from viruses, but these viruses are edited so they don't have the infectious disease, they just assist in the process. And so two main vectors are the viral vector and the non-viral vector. And just some physical and chemical approaches are needle injections, gene guns, or chemical disruption. Now, there are a lot of pros and cons to human gene therapy. So one of the pros is that it provides positive effects for future generations. As mentioned for germline, you can reduce the effect of having a disorder being passed down to your future children. And it also gives hope for a lot of healthy lives for future patients. And because this human gene therapy, once it's inserted, it can just change your life rapidly, just like that. And it also has long lasting effects because it has the potential to get rid of this person's symptoms for life. And it only has to be given one time. It's a one dose for the human gene therapy, whereas other treatments, they require multiple ones. And lastly, it's a rapidly changing technology. It's constantly evolving and growing with its uses, and we're learning so much from it. And hopefully the FDA, we would get more approvals for certain human gene therapies to be used. Now, as for the cons, there are ethical issues and it could be potentially be dangerous since it is relatively new still. Hence, it is experimental. So there's a lot we don't know. It could be risky, it could be not. So it's a 50-50 chance. And lastly, it's very expensive with all this research and experimental going into it. And one of the most costly prices is this spinal muscular atrophy medicine, which costs $2.1 million just for one course of treatment. So there's a lot of pros and cons to weigh out with human gene therapy, but it's a rapid development and evolving method that has benefited a lot for our society. And thank you for listening.